Hi, my name's Rachel, and I'm going to be presenting about a previous project that I worked on, and it is basically involving the increase of purity in ventricular myocyte populations. So basically, currently, there's talk about turning pluripotent stem cells that are undifferentiated into cardiomyocytes or heart cells to help therapeutically heal someone who had undergone some sort of injury that would cause the heart to be damaged, so maybe like a heart attack, something like that, basically. So basically, the current method is you have pluripotent stem cells, you add a chemical, get some progenitors, and then you add more chemicals and get a population of cardiomyocytes. But unfortunately, the current methods, which is called the GIOE protocol, creates a population that is not homogeneous. It creates atrial cells, ventricular cells, and nodal cells. And basically, if you try to use this population for a therapeutic application to heal someone, it would not work out because all the different cells will actually lead to further problems. It usually causes arrhythmias or irregular pacemaker activity, which is obviously not what you want. So my project was basically to generate a homogeneous population of ventricular cardiomyocytes by modifying the GIOE protocol, which is what is currently used. And the reason we chose ventricular cardiomyocytes is that typically when someone undergoes a heart attack, which is usually when someone would have to undergo some sort of therapy for healing their heart, usually the ventricular part of the heart is what's damaged. So that is why we're focusing on ventricular cardiomyocytes. I'm going to talk about the plan in the next slide, but basically, once we produce this population, we also came up with a new way to identify if the population is mainly ventricular cardiomyocytes, because the current methods for identifying these cells actually end up destroying the cells themselves. Usually the two markers that people look at are IRX4, which is a nuclear marker, and MLC2V, which is a myosin light chain subtype. And we focused on MLC2V. We used CRISPR-Cas9 to basically insert a GFP protein right where the MLC2V would be expressed so that GFP would be expressed when that is expressed. So hopefully when MLC2V is expressed in the ventricular cells, the ventricular cells will fluoresce, so we will know that those are ventricular cells and not some other type of cardiac cell. So basically our experimental design was basically drawn from different papers that we read, where they basically found that retinoic acid was involved in a lot of the cell fate, turning the cells into ventricular cardiomyocytes once they matured. So we had a couple different controls. We had the negative control, which is the typical GIOE protocol, where you add a certain chemical from day 0 to 1, a certain chemical from day 3 to 5, and that's pretty much it. And this produces the heterogeneous population, which is not what you want. We have the experimental condition, which is what was found in previous papers that they actually carried out and tried to get a more pure population, which is when they added retinoic acid from day 0 to 1. And they did not get a very pure population, so we were going to try to basically modify that and ensure that, we can get a more pure population. Maybe they just weren't picking the right temporal frame for adding the retinoic acid. So what we were going to try to do was add retinoic acid from day 5 to 8, and we chose this based on literature saying that this is basically when the body or when retinoic acid would be involved in determining the cell fate. So that is why we chose that time frame, and basically we would add that from day 5 to 8 and then add it to the media in the cell, and then once the cells matured after 30 days, we would harvest them for flow analysis, where we could determine if the cells in the population were ventricular cardiomyocytes. And this would be done using flow analysis, as I said, where we would look for CTNT, which is a cardiac marker, and we would look for MLC2V, as I discussed before, which is the ventricular marker, which we will be able to identify using the CRISPR-Cas9 technique. So hopefully by using this way, we will be able to determine if the population will become more pure using this method. So we'll see how that turns out. Thank you.